Obesity is bad for heart health, right? In this video, we're going to discuss a scientific research paper that found that communication between fat cells and the heart can lead to some unexpected outcomes. Hey there, welcome to Science is You. Brief disclaimer, there are many factors that contribute to health and everyone is unique. Although research can help us understand mechanisms of disease and discover new therapies bit by bit, nothing in this video should be taken as specific health advice. Obesity is generally linked to health problems like diabetes and cardiovascular disease. But why is this? Excess fat is stored in cells called adipocytes. These cells have numerous roles in the body and secrete substances that can affect other organs, causing changes, for example, in sensitivity to insulin and inflammation. However, when adipocytes contain too much fat, they can't function properly. Specifically, excess fat stresses mitochondria, which are structures within the cell that produce energy. Cells need this energy to grow and survive. So, does that mean if an adipocyte takes in too much fat, it just dies? No, cells can adapt and change to compensate for their environment. Actually, a brief, mild stress can be good in some cases because it stimulates the cell to start adapting in a way that protects it from future, perhaps more lethal stresses. However, if cells are overwhelmed by a strong or prolonged stress, the cell can't compensate. Imagine if you're training to run longer distances. If you run for 10 minutes a day for several days, you might have an easier time running 20 minutes the next week. But if you go out at the hottest part of the day and try to run for an hour, you'll probably pass out. Likewise, large amounts of excess fat can overwhelm adipocytes, resulting in damage to their mitochondria. Damaged mitochondria can release molecules called free radicals that go on to damage other molecules in a process called oxidation. You're probably familiar with oxidation of metal to rust. Similarly, free radicals in cells can damage molecules like DNA and proteins. So there's a lot of interest in counteracting this process, for example, through consumption of antioxidants and figuring out what causes excess free radicals. In this paper, the researchers used a mouse that had a genetic change which caused mitochondrial stress in their adipocytes. When fed a diet high in fat, these mice mimicked the mitochondrial damage observed in obesity. Previously, the authors of this paper observed that not only did these mice show oxidative stress in adipocytes, but also in the heart. This suggests that some sort of pro-oxidation signal was being transported somehow from the adipocytes to the heart. The authors hypothesized that the signal was transported from the adipocytes to the heart within extracellular vesicles, which are small enclosed pouches that pinch off from cells and can be reabsorbed by other cells. These vesicles carry contents from the cell, which can consist of many different proteins, enzymes, signaling molecules, and cellular structures, which can even include parts of mitochondria. Here, the authors found evidence that obesity can trigger adipocytes to release damaged mitochondria enclosed in extracellular vesicles, which travel through the bloodstream and can be taken up by cells of the heart. Specifically, they observed that their mice, which mimicked obesity on a high-fat diet, had more circulating extracellular vesicles than normal mice. Extracellular vesicles can be measured by counting them directly using a special microscope-like equipment and measuring the amounts of proteins that are known to be contained in extracellular vesicles, such as CD63 and CD81. The vesicles could be further traced to adipocyte origin by measuring increased levels of proteins like FABP4, which are specifically produced by adipocytes. To see how extracellular vesicles from adipocytes affect cells of the heart, the authors collected vesicles from the mice. This can be done by filtering a blood sample to get rid of blood cells and larger components, then spinning the liquid at high speed to pull the small extracellular vesicles to the bottom of the tube. They then put the vesicles onto cardiomyocytes growing in a dish and measured levels of oxidative stress using a chemical that changes color and glows fluorescent when it is oxidized. Extracellular vesicles from their mouse obesity model cause more oxidative stress than vesicles from healthy mice. More oxidative stress was also observed in cardiomyocytes treated with extracellular vesicles from healthy mice that were fed a high-fat diet. Interestingly, cardiomyocytes treated with vesicles from healthy mice had lower oxidative stress than cardiomyocytes that were not treated with any vesicles at all. The authors next wanted to see whether vesicles from adipocytes are indeed taken up by the cardiomyocytes. To do this, they grew adipocytes in the lab and collected their extracellular vesicles. They then stained these with a dye and treated the cardiomyocytes with them. They saw that the cardiomyocytes now contained the fluorescent dye, indicating that they had taken up the vesicles. They further observed that vesicles collected from adipocytes with stressed mitochondria caused significant oxidative stress in the cardiomyocytes. This experiment showed that cardiomyocytes can take up vesicles from adipocytes in a dish, but what about in the body? To study this, the researchers injected the vesicles into normal healthy mice. They observed that the vesicles were indeed taken up by cells in the heart. Vesicles from healthy, non-stressed adipocytes did not cause any oxidative stress in the heart, which is what they observed in the dish. However, when they exposed the adipocytes to high levels of fat, which stressed them, and simultaneously fed the mice a high-fat diet at the time they injected the vesicles, and significantly higher levels of oxidative stress were found in the heart. In addition, mitochondrial function was decreased. This effect, however, was transient, lasting only about an hour. Surprisingly, later on, some hours after the injection, oxidative damage was significantly reduced, as was mitochondria-specific oxidative stress. This suggests an adaptive antioxidant response was triggered somehow by the vesicle injection. Further, some mitochondrial functions were enhanced. These observations suggested that extracellular vesicles from stressed adipocytes might enhance oxidative stress in cardiac cells temporarily, but the cells rapidly adapt to restore and enhance mitochondrial functions. To understand how this happens, the authors examined what proteins were contained within the vesicles. They found a lot of mitochondrial proteins. Further, they found that vesicles from adipocytes exposed to high fat or collected from obese mice contained more mitochondrial proteins than those from healthy mice. The authors also measured indicators of mitochondrial function, like oxygen consumption in the vesicles, which fluctuated predictably with the addition of chemicals that impact mitochondrial function. This suggests that functioning mitochondrial components are somehow present in the vesicles. This could happen when these vesicles bud or pinch off directly from the mitochondria in response to oxidative stress. To investigate these mitochondrial-derived vesicles, the authors used a mouse that was genetically changed to produce mitochondria with a tag that could be detected by a fluorescent dye under the microscope. Importantly, the tag is only in mitochondria of adipocytes. However, after some weeks of feeding these mice a diet high in fat, they found the tag in the bloodstream and heart of the mice, indicating that adipocyte mitochondria in some form had been transported. Further, they observed the tag signal specifically in cardiomyocytes. If these vesicle-transported mitochondrial parts from the stressed adipocytes integrated into the cardiomyocytes, it could help explain the temporary malfunction of cardiac mitochondria previously observed. Importantly, this mitochondrial transport from adipocytes was only observed when the mice were fed a high-fat diet for 11 weeks, but not for only a few weeks. But do these vesicles play a role in cardiovascular disease? To determine this, the authors took extracellular vesicles from adipocytes that had been stressed by exposure to high fat and injected them into mice. They then simulated a heart attack on the mice by briefly stopping blood flow through an artery. Surprisingly, mice that had received the vesicle injection had reduced cardiac damage compared to mice that hadn't had them. The authors conclude that together these observations suggest that counterintuitively, the transfer of vesicles from stressed adipocytes to cardiomyocytes doesn't cause cardiovascular disease, but rather counteracts it. To figure out whether these processes also happen in people, the authors collected extracellular vesicles from blood samples of lean and obese individuals. Obese individuals who were also metabolically unhealthy had significantly more extracellular vesicles in their blood than lean individuals. Extracellular vesicles from obese individuals contain more mitochondrial proteins and oxidized proteins compared to lean individuals as well. Based on all these experiments, the authors speculate that in obesity, adipocytes respond to intense stress from excess fat accumulation by releasing extracellular vesicles that carry parts of oxidatively stressed mitochondria. The vesicles can then make their way to the heart, where they are taken up by cardiomyocytes. The oxidative stress induced in the cardiac cells triggers them to adapt and respond in a way that makes them more resistant to injury from lack of blood flow during a heart attack. However, the authors stress that there's likely an upper limit beyond which the heart cells can't compensate and instead are harmed by the oxidative stress. Further research is needed to uncover what this limit is and how this knowledge might be applicable in clinical settings, where many additional factors influence the relationship between obesity and overall health. Although this study focused on the cardiovascular system, it's interesting to note that the authors observed substantially more uptake of adipocyte vesicles in the liver. What do you think some clinical implications of this study might be? Apart from obesity, could extracellular vesicles from stressed adipocytes, like the authors used here, be used eventually as a clinical therapy to protect against heart damage? What additional experiments or tests would you want to see before something like this would be considered? Drop your ideas in the comments. If you like this video, don't forget to subscribe to help this channel get off the ground. And remember,